Hello everyone and welcome back. So today we are going to continue and talk about the genetics and genomic basis of sleep. So the goal of today's presentation is to cover all the topics listed here: genetics of sleep, sleep disorders, why study sleep, genomic basis of sleep, genetic basis of sleep stages, the importance of sleep, function of sleep, genes, circadian clock, personalized medicine, ethics in sleep, and future of sleep genetics. So I'm going to present two questions and then we go back to the topics and then we come back and discuss these questions. So question number one is the case number one, which is a 23-year-old woman with a presentation of restless leg syndrome. She is curious about the risk of her passing on to her newborn baby. The baby's father also had the same diagnosis of restless leg syndrome. So what is the risk factor for the baby to inherit the disease? So that's one question. The second question is a 13-year boy who presented with gradually worsening insomnia, leading to an inability to sleep more than 4 hours. So this boy was 13-year-old and it got worse and worse insomnia and he was not able to sleep more than 4 hours in 24 hours. The clinical feature of this patient included weight loss, balance problems, psychiatric symptoms of anxiety, depression, and mood disorders. He passed away in 8 months from the rare genetic sleep disorder. So sleep is a fundamental physiological process which is essential for our physical and mental well-being. While environmental and lifestyle factors like stress, diet, and routine are commonly recognized as factors which influence sleep, recent scientific research has revealed that sleep is also deeply rooted in our genes. Sleep genetics and genomic basis offer a new insight into how sleep is regulated, why individuals have different sleep patterns, and how sleep disorders might arise. So the 24-hour cycles known as circadian rhythm that dictate sleep and wakefulness are the heart of the sleep regulation. These rhythms are controlled by our biological clock in the brain's suprachiasmatic nucleus. This suprachiasmatic nucleus, also known as SCN, responds to environmental cues like light, darkness. So when there's light outside or there's a darkness inside, and then adjusts sleep pattern according to that light outside the body. However, suprachiasmatic nucleus' ability to regulate sleep is supported by the set of core genes known as clock genes. So the clock genes, which include clock, BMAL1, PER, and CRY, PR is period and CRY is cryptochrome, are crucial in regulating circadian rhythms of human beings. These genes operate in a feedback loop that cycle every 24 hours. So this feedback loop, which um, loops in every 24 hours, it creates a rhythm that influences our sleep and other physiological processes such as the release of different hormones in the body and basic metabolism. For example, PER and CRY genes inhibit their production by interacting with clock and BMAL proteins, and in this way, they create an oscillation that drives the sleep-wake cycle. So sleep-wake cycle is a cycle which is 24-hour cycle controlled by a circadian rhythm. So when there's a variation in these genes, also known as mutations, it can significantly affect our sleep. So a well-known example of mutation is PER3, PER3, and this PER3 gene has been linked to advanced sleep phase disorder, ASPD. So this ASPD, advanced sleep phase disorder, is a condition where the individual or patient have unusually early onset and wake time. So these patients, when this gene is present, the patient go to sleep early and they wake up late. So they cannot go keep awake for a very long time and they cannot sleep for a long time or later in the morning. So they have early onset of sleep and they have early wake onset. On the other hand, when there is a mutation in CRY genes, that has been associated with delayed sleep phase disorder. So delayed sleep phase disorder is DSPD, where people struggle to fall asleep and wake up conventional time. So they have a difficult falling asleep, but they wake up in conventional time. So one is advanced sleep phase disorder, where they sleep early and wake up early, and then have delayed sleep phase disorder, where they wake up, they cannot go to sleep early. So when we are studying the field of genomics, this is a field that examines the entire genome rather than just single genes. And this has expanded our understanding of sleep even further. So genome-wide association studies, or GWAS, has identified several genetic loci associated with sleep traits such as sleep duration, quality, and timing. So the studies, GWAS, which is also known as genome-wide association studies, they have identified several genetic location of the genes, and these location on the genes, they are responsible for the duration of the sleep, varying the duration of sleep or the quality of sleep and the timing of sleep, early onset or late onset. These studies reveal that sleep is polygenic, means multiple genes contribute to the sleep regulation. So the sleep is not controlled by only one gene, but there are multiple genes and multiple genetic factors that are responsible for the sleep quality. One prominent discovery from these genomic studies is identifying a gene which is known as ABCC9, which influences sleep duration. So this new identification of a gene ABCC9 is especially responsible for influencing the sleep duration of the sleep. Variants in this gene have been associated with shorter or longer sleep in human. So these are different variation in this gene, and this is affecting the duration of the sleep. So that variation can either cause a shorter sleep or longer sleep. So this demonstrates that even slight genetic difference can lead to significant variation in how much sleep individual can need or how much sleep a person will have. Other genomics insights have highlighted the role of genes involving in regulating neurotransmitters, and these neurotransmitters which are affecting our genes are serotonin, dopamine, and GABA, gamma aminobutyric acid. And these neurotransmitters are crucial for promoting sleep or promoting wakefulness. For example, when a gene is these neurotransmitters are promoting sleep, the person will go to sleep, and when they are promoting wakefulness, the person will wake up and will not be able to go to sleep. For example, variation in ADRB1 gene that will affect the activity of adenosine transmitter, which is a sleep promoting molecule. So, and that has been linked to variation in sleep patterns. So, adenosine is a sleep promoting molecule. So when that is affected, that will result in either sleep promotion or sleep deprivation. Additionally, a mutation in DEC2 genes has been found in people who are required less sleep than the average person, and this indicates the genetic basis for short sleepers. So those people are known as short sleeper because they cannot go to sleep for a long period of time. So genetic research has also provided a deeper understanding of sleep disorders, different conditions which affect our sleep. The one of the most common is insomnia, which is unable to sleep; restless leg syndrome, which is kicking or sudden kicks of jumping of the leg during the sleep; and narcolepsy, which is going to sleep in the morning all day and unable to keep awake. And then you have cataplexy, which is going to sleep very quickly, and with a muscle loss of muscle, patient can drop there. So, for instance, the narcolepsy is a disorder that it is characterized by excessive daytime sleepiness. So, the person with narcolepsy will have excessive sleepiness all day. Even they are sleeping at night, but they still in the morning they will still sleepy. And if they have sudden loss of muscle control, in addition to that, that will a disease which is known as cataplexy, and that has been linked to variation in HLA gene complex, which is human leukocyte antigen gene, and that plays a role in immune system regulation. So this is another image which presents a conceptual framework for understanding the regulation and function of sleep through a multidisciplinary approach. It is developed by University of Surrey. It visually connects the key elements which are involved in sleep mechanism. The core of the framework is sleep quality, which is influenced by multiple factors such as circadian rhythms, which is shaped by light dark cycle, and with images of Earth and city lights, Homeostate, which tracks the sleep-wake's history, the history of sleep and wakefulness, social and behavior influences, which affecting sleep patterns, the cognition and health, which are linked to sleep quality, aging, which is shown as determinant of sleep regulation, genetic variation, which is depicted at the bottom, which influence the sleep mechanism. This framework underscored the complexity of sleep regulation, integrating biology, environment, behavior, and health. This served as an insightful reference for sleep research and neurophysiology. So let's look at another image, and which is a presentation slide from University of Surrey, how multidisciplinary clinical research centers. It outlines the key features of MHRA accredited phase unit 1, emphasizing the multi-level assessment and quality control condition for research. The main feature are participants' residency. Research subjects stay in the facility for days, weeks, or months. Multi-level assessment of these subjects by electrophysiology, sleep, and awake cycle. Recording of their timings, cognition, using 1 bag, 2 bag, and 3 bag cognitive test. Biomarker, which are blood, saliva, urine, and adipose tissue test. Then controlled environment in these conditions for precise regulation of temperature, light, and diet. High throughput capacity is 6 to 12 participants can be involved for the study at the same time. Additional resources for bioinformatic and statistical analysis were done, and healthcare. So we can see healthcare personnel working with patient in clinical setting, and the electrophysiological data is displayed on handheld device for remote monitoring, and cognitive test diagram illustrating memory-based challenges. So this image is showing 1 night of polysomnography, which is a sleep study. PSG, polysomnography is a sleep study done in 1 night of 5 to 8 hours, and present a visual presentation of sleep architecture over 8-hour period for 22-year-old male. This study tracks sleep stages, including EEG pattern transition throughout the night, and the sleep stages are a wake period that occurs intermittently. Patient wakes up in the middle of night and then go to sleep. REM sleep, which cycles appears to various intervals, and then we have stage 1 to 4 represent night light to deep sleeps as the sleep awake sleep cycle. So it's important to remember in old pictures, old diagram, the stages of sleep were four. Non-REM sleep is what stage 1, 2, 3, 4. But the new classification, the non-REM sleep is only 3 stages. So stage 3 and 4 are combined. So you should not see stage 4 in all new data. So final awakening, which is marked by the 8-hour mark, so where the patient is or the subject is awaked and the study is complete. The EEG data shows the light sleep, shows sleep spindle, different and deep sleeps or sleep SWS highlights slow wave activity. So when the person is going from awake to light sleep to deep sleep to REM sleep, the wave EEG waveform gets slower and slower from beta to the alpha to theta and delta, and then patient can wake up and we can tell with a different marker which stage of the sleep the patient. So the time since the lights are on is x mark from 0 to 8 hour. Sleep stage projection on y-axis. Y-axis is showing different stages of sleep, and EEG reading are visualizing waveform pattern. And so this polysomnography chart provides an insight into sleep cycle pattern and brain monitoring, making it relevant to neurophysiology and sleep research. So disruptions due to insufficient sleep and different times every day changes the expression of many genes. This image is a scientific presentation slide from University of Surrey explaining how insufficient and missed time sleep affect the body's biological process. Why does sleep matter? So sleep affect how the blood body functions at a genetic level. Changes in sleep pattern can disrupt our circadian rhythm, which controls sleep awake cycle, and also what happens with too little sleep. The left side of the image compares sufficient sleep versus insufficient sleep. Researchers studied people who slept 8 hours versus 6 hours per night and measured how sleep deprivation impacts their blood transcriptions, gene expressed in the blood. So trans blood transcriptome is the gene expressed in blood. Findings show that less sleep weakens the body rhythm, affecting health over time. So the next question is, how does missed time sleep affect us? The right side explains how irregular sleep pattern interferes with gene regulation. In this study, participants followed unnatural sleep cycle to observe changes in their body biological clock. The graph showed shifting patterns in gene expression, proving that missed time sleep disrupt natural body function. Why is this important? Because the understanding these effects help researchers develop strategies to improve mental health, performance, and overall well-being. Now, this image is illustrate the effect of missed time sleep on rhythmic gene expression. The study shows the six-fold reduction in rhythmic gene activity when sleep is misaligned with the body natural clock. So the main topics or main finding of this study were that heat map comparison, which is the left heat map in phase, present normal gene expression during proper sleep-awake cycle, and on the right heat map, which is out of phase, shows disrupted gene pattern when sleep is missed time, causing irregular activity in biological rhythm. The second, the rhythmic gene reductions in normal sleep in phase 13, 1396 genes exhibit rhythmic cycle, which is 6.4% of the total recordings. In missed time sleep, out of phase, only 228 genes maintain rhythmic activity, which is 1% of the total. The circular plot analysis on the right, a circle graph is compares the gene expression pattern across different sleep phases, which highlighting the disruption caused by misaligned sleep schedule. So the reason it's important is that proper sleep timing regulates essential biological function, including metabolism, immune response, and cognitive health. Sleep disruption can alter our gene expression, potentially leading to health complication over time. And the possible application of this study is relevant to neurophysiology, sleep science, and machine learning in medical monitoring. Here we see the same results, and this image explain the main effect of sleeping out of phase on gene regulation. It highlights how altered sleep timing affect various metabolic and molecular processes. Researchers analyzed 31,905 transcripts to study gene expression changes due to misaligned sleep. 913 genes were down-regulated, while 206 genes were up-regulated when sleep occurred outside the natural rhythm. The affected biological processes include macromolecular metabolism, gene expression, nucleic acid metabolism, RNA metabolic process, DNA and RNA binding, hemoglobin metabolism, oxygen transporter activity, um, uh, peroxyredoxin activity, which is related to cellular stress response. And the network diagram shows that gene interaction network, visualizing how different genes are connected. And the key genes were ESR1, CUXI1, and PIK3CA, and these are labeled here, which highlighting their roles in regulating metabolism and cellular function. So here is another image which explores sex differences in shift work and their implication on accuracy based on circadian rhythm. So there are two heat maps, left side and right side. The left side represent male accuracy and right represent the female accuracy during shift work. Both graphs compare accuracy based on circadian phase. 0 is degree is equal to melatonin onset and relative clock hours from 0 to 24 hours period, and time awake varies from 3.1 to 18.16 hours. The color scale interpretation, the red is minus 1.2, lowest with the lowest accuracy. The orange is minus zero point 0.9, yellow 2.6, orange to yellow, which is 0.9 to 0.6, which is moderate accuracy. The green one is minus zero point 0.3 to blue 0, higher accuracy, and dark blue is 0.3 with the highest accuracy. Shift period, day shift marked with the white line, and night shift is marked on the red line on the right side. So day shift and right shift, you can see the white line and the right on the left side and the red line on the right side. These lines show where accuracy fluctuate based on working hours. So the data reveals how circadian rhythm influence accuracy differently for men and women. And women, it has implications for work performance, cognitive efficiency, and health risk in shift work. So understanding these patterns helps optimize work schedule and fatigue management categories. So the genomic basis of sleep is complex and poorly understood. The circadian rhythm and neurotransmitter effect are genetically controlled. Mutation in some genes reduces response to SD, and twin studies, monozygotic and digotic studies have been done, and single gene disorders and HLA human. So sleep is ubiquitous and complex behavior in its manifestation and regulation. Despite its essential role in maintaining optimal performance, health and well-being, sleep genetic mechanism remains poorly understood. Despite increasing studies and mining, large databases, a coherent picture of sleep genes have yet to emerge. We highlight the result of unbiased genetic screens, mainly in humans, mice, and fruit flies. We emphasize normal sleep and refer to lessons learned from circadian field. So through genetic analysis of sleep, we hope to learn about molecular substrate of its remarkable behavior and its pathologies. Sleep is remarkable because we spend a considerable portion of life in this stage. On the average, more than 24 years of most of us, we know little about its likely numerous function. Clearly, sleep is important as lack of sleep, poor quality sleep, and sleep disorders greatly impact performance and well-being and may lead to poor health. Genetics can be used to approach a variety of questions about sleep regulation, sleep function, abnormal sleep, and consequence of poor or insufficient sleep. For example, the research aimed at discovering the gene variants altering susceptibility to a deleterious effect of sleep loss and the molecular pathway activated by sleep loss is of great interest from both the basic scientific understanding and the clinic point of view. So sleep geneticists look at the astounding progress made in the closely related circadian rhythm research field, and the result of these mutagenesis screens in flies and mice for circadian phenotypes, mostly variations in circadian period, led to discovery of core set of interactive genes, that is clock genes that pace the clock and to a Nobel Prize in 2017.
Good morning. Good morning to Nobel Forum and this press conference for the announcement of this year's Nobel Prize in Physiology or Medicine. My name is Thomas Perlman and I'm the Secretary of the Nobel Assembly. I will first read the announcement in Swedish, followed by English, and we will then present some background to the prize and open up for questions. Good morning. Good morning to Nobel Forum and this press conference for the announcement of this year's Nobel Prize in Physiology or Medicine. My name is Thomas Perlman and I'm the Secretary of the Nobel Assembly. I will first read the announcement in Swedish, followed by English, and we will then present some background to the prize and open up for questions. Nobelförsamlingen vid Karolinska institutet har idag beslutat att Nobelpriset i fysiologi eller medicin år 2017 skall delas lika mellan Jeffrey C. Hall, Michael Rosbach och Michael W. Young för deras upptäckter av molekylära mekanismer som styr cirkadisk rytm. The Nobel Assembly at Karolinska Institutet has today decided to award the 2017 Nobel Prize in Physiology or Medicine jointly to Jeffrey C. Hall, Michael Rosbach, and Michael W. Young for their discoveries of molecular mechanisms controlling the circadian rhythm. Here are the three laureates. Jeffrey Hall was born in New York and performed his seminal work at Brandeis University. He's now retired and lives in Cambridge in Boston. Michael Rosbach was born in Oklahoma City and performed his prize-winning studies also at Brandeis University, where he's still on the faculty. And finally, Michael Young was born in Miami and did his work at Rockefeller University in New York, where he also remains on the faculty. I will now describe the discoveries behind this year's award. So this year's prize concerns the circadian rhythm. The word circadian originates from the Latin word circa, meaning around, and dies, meaning day. But in a way, it is actually about astronomy. Ever since the emergence of life on Earth, about 4 billion years ago, evolving life forms had to adapt to the rotation of our planet. This ability to prepare for the regular daily fluctuations is crucial for all life forms. But how is this possible? This year's Nobel laureates have been studying this fundamental problem and solved the mystery of how an inner clock in most of our cells in our bodies can anticipate daily fluctuations between night and day to optimize our behavior and physiology. This existence of an inner clock was first described already in the 18th century. The French astronomer de Meuron studied the mimosa plant that opens its leaves towards the sun during day but closes at dusk. De Meuron asked what would happen if the plant was exposed to constant darkness. And he found that even then, for several days, the leaves continued to follow their regular 24-hour daily rhythm. In the 20th century, it was established that also other plants and animals, including rodents and humans, have their own biological clocks following the daily circadian rhythm. Studies of the clock in fruit flies show that many different behaviors are controlled by the clock. Fruit flies are really excellent model organisms for studies of genetics, and in the 1970s, a mutation that affected the fly's daily rhythm was identified. The mutated locus was called period. But how did the mutation affect the internal clock? This year's laureates, they wish to peek inside the clock and figure out how it actually works. A first breakthrough came in 1984 when Jeff Hall, in collaboration with Michael Rosbach and Michael Young working independently, were able to isolate the period gene. Now the scientists could begin to disentangle the cellular wheels and cogs of a clock machinery. Jeff Hall and Michael Rosbach showed that the period protein itself was cycling and peaked during night and was low during day. In 1990, they showed that the period mRNA also oscillated, as shown on this slide, and accumulated several hours before the period protein. These and other important observations led to a brilliant idea for how the clock works by a feedback control mechanism. The idea was shown to be correct, and in the next few slides, I will show how it actually works. When the period gene is active, period mRNA is made. The period mRNA is transported to the cell's cytoplasm, where period protein is made. When period protein accumulates, the protein moves into the cell's nucleus, where it blocks the activity of its own gene. When the gene is blocked, no more RNA can be made. Period protein levels will decrease, and the gene will no longer be blocked. And the production of mRNA can start all over again, and the cycle can continue. But it remained unclear how the period protein could accumulate in the nucleus to inhibit the gene. This problem was solved when Michael Young discovered the second gene, called timeless that is also critical for the clock. This is shown in this experiment. The top diagram shows how period mRNA is cycling over several days in normal flies. The bottom diagram shows that the cycling was disrupted when the timeless gene had been inactivated. But how did the timeless protein affect the period gene? Additional experiments show that timeless protein bound to the period protein, and together they entered the cell nucleus where they could inhibit the period gene. It was also critical to understand how the speed of the oscillations can be controlled. Michael Young identified yet another protein called double time, or DBT. This protein was shown to slow down the accumulation of the period protein, which explained how the oscillation can be adjusted to more closely match a 24-hour cycle. Jeff Hall and Michael Rosbach showed that two additional proteins, clock and cycle, activated the period gene in fruit flies. So, with all these groundbreaking discoveries by the three laureates, an elegant feedback control mechanism for the biological clock had finally been unraveled. We now recognize that biological clocks function by these same principles in other animals, including in our cells, in humans. Most of our cells in our bodies have these clocks. Our inner clock adapts our physiology to the dramatically different phases of the day and regulates many critical functions such as behavior, hormone levels, sleep, body temperature, and metabolism. We do not feel well when there is a temporary mismatch between our external environment and the internal biological clock, for example, when we experience jet lag. Studies have also indicated that chronic misalignment between our lifestyles and the clock is associated with increased risk for various diseases. Since the paradigm-shifting discoveries by Hall, Rosbach, and Young, circadian biology has developed into a highly dynamic research field with vast implications for our health and well-being. Here they are again, Jeffrey Hall, Michael Rosbach, and Michael Young. This is the end of the presentation, and I would now wish to present our expert panel, who would be happy to respond if there are questions. So, at the far end, we have Professor Julien Sirat, who is a member of the Nobel Committee. In center, we have Anna Wedell, who is the chair of the Nobel Committee. And on her side, we have Professor Carlos Ibanez, who is also a member of the Nobel Committee. So please, if there are any questions, please go ahead. There must be at least one question. Okay, please go ahead.
Have you already been in contact with the professors? Did you dare to call them in the middle of the night?
We are usually not afraid of calling them about this. And I was able to get hold of Michael Rosbach and Jeffrey Hall, but I haven't reached Michael Young. So they just got the news.
How did they respond?
I first got hold of Michael Rosbach, and he was silent, and then he said, "You are kidding me." Is there any other question?
Yeah, I'm
Yeah,
curious
please go ahead.
about the dynamics of this system. So if one were to change the cycle to like 6 hours instead of 24 hours for humans, for some reason, or chicken, they are being kept in very short cycles. What happens with this system? Does it adapt to very short?
I think I'll ask Anna to respond to that question.
Well, we know that there are, in rare cases, there are humans who have a little bit altered length of the period, not as you described 6 hours, but a little bit shorter, and they get sleep problems. We don't know of any drastic disruptions in humans of the clock. There are, of course, animal experiments, and if you disrupt the clock in animals, you will get effects on metabolism and on various physiological parameters. But in humans, it's not really possible to do those kinds of experiments.
But are there any treatments or possible treatments for sleep disorders that could be based on this finding?
Well, an obvious answer would be melatonin, which is a hormone that is secreted before we go to sleep, and that it can also be used to for those kinds of problems. And some people even use it for jet lag. And also, if you're blind, for instance, your rhythm will still be going, but it will be out of sync with or misaligned. It will be free running, since it will not be entrained by light. And then you can use similar compounds to melatonin to try to adjust that. Did you hear me?
Here's one question. Please go ahead.
Hello, I'm from Chinese media, Green Post and China Radio International. My understanding of this prize is in physiology, right? So can you tell us how long have you, or how did you find them, and what is their contribution to mankind?
How we found them and their contribution.
I'll ask Carlos Ibanez to respond to that question.
How we found them. Our mechanism is based on nominations from different parts of the academic community and institutions, universities, other scientists. And then these scientists have been nominated by these other members of the academic community and research community. And then that's how we found them. Our work is directed by the nominations that we get. We are aware of interesting fields, but we can only follow up nominations from the scientific community. And then we investigate whether the discoveries merit this prize, and that's how we found them. The contribution to humankind is the discovery of a fundamental mechanism underlying very important aspects of physiology, how our cells can keep time. Did I answer your question?
Okay, here's one question.
I've got one question. I'm Fred from China's National Xinhua News Agency. Regarding to this year's discovery, do you think this discovery by the scientists can solve the problems, especially that we encounter in the Nordic countries where you've got long, dark winter days that people tend to have depression? And is this discovery actually shedding lights to solving the problem to boost, people's energy?
Could Julene ask?
So I guess the question was how this discovery might solve the problem of how people cope with extreme changes in darkness or sunlight. And I think what it can do is raise awareness to the importance of proper sleep hygiene and the importance of really making certain that we allow ourselves to, go to bed at an hour that's suitable. I guess for many of us, it's around, between 10 o'clock or so, even if the sun is shining on Midsummer Day. And it probably helps us to be aware of the fact that we need to be in a dark environment to get the best sleep. And that we shouldn't be afraid of using our alarm clock, even when it's the darkest times of the year. So I think what it can do is help raise awareness.
Okay, is there any else who is interested in asking a question?
Is it possible for you to say it in Swedish? Kan du prata svenska och förklara?
Det går bra. Så att frågor på svenska går bra också. Eller andra språk, om vi klarar det.
Kan ni förklara då texten på svenska?
Ja, alltså det skulle jag föredra att göra i intervju sen, men alltså det handlar ju om en inre biologisk klocka som man har känt till och som går oavsett om det är ljust eller mörkt, om det är dag eller natt. Den är inre, den går av sig självt. Den ställs rätt av yttre faktorer som ljus. Men den fortsätter att gå och den påverkar väldigt mycket av vår fysiologi. Och de här forskarna har förstått mekanismen för hur den här klockan fungerar i våra celler. Klockan finns i nästan alla våra celler i kroppen och är väldigt viktig för väldigt många processer. Okay. If there are no more questions to ask, I know many of you are looking for interviews, so we can go ahead with that. So with that, I would like to close this conference and thank you all for coming. Thank you.
Professor Anna Widell, Chair of the Nobel Committee, could you describe to us what is circadian rhythm?
So circadian, it derives from the Latin. So circa is around and day is day. And this is the daily rhythm we have to anticipate and adapt to the dramatically different conditions during day and night that arise from the rotation of the Earth.
So the three laureates, what problem have they solved?
So it had been known for a long time that there are diurnal or circadian rhythms, but how it worked wasn't known at all. And it was debated for a long time whether it was a response to external signals or something endogenous. And before they started their work, it was understood that it was endogenous, and it was even understood that single genes could influence the circadian rhythm, but nothing was known about the mechanism. And there were different hypotheses, but they found out the mechanism that controls the machinery of the clock.
So if we
So regarding the importance of sleep, it is essential for cognitive function, physical health, and emotional well-being. Inadequate sleep is linked to various health problems, including obesity, cardiovascular disease, and depression. So sleep help in memory consolidation, especially stage 3. And stage 3, if you don't go to stage 3 or non-REM sleep stage 3, you will not have a good memory consolidation. So whatever you're trying to memorize and learn, you'll not be able to do that. So good sleep is good for memory consolidation. Sleep helps stabilize and enhance the memories. The immune function of the body. The sleep is boost the immune system's ability to fight off infection. Sleep also regulate hormone like insulin and leptin, which control the hunger and metabolism. So it helps in metabolic regulation, immune function, and memory consolidation. So in this table, the genes investigated to contribute to genotype-dependent differences in diurnal preferences, sleep timing, sleep and EEG, sleep structure, and sleep duration, and sleep hormones, in circadian, and then the neurotransmitters, adenosine, and the other neurotransmitters and potassium channels, and then immune system. So this image represents a comparison of brain activity and genetic influence between monozygote twins and dizygote twins using EEG data. So the part A displays 4 brain images with color-coded regions showing the degree of correlation or square between brain areas. Monozygotic twins brains show more red and yellow, indicating higher correlation, while dizygotic twins (DZ) show more blue and green, indicating lower correlation. A label F and W point to specific regions under analysis. Part B, the second part, the bar graph comparing inter-class correlation coefficient (ICC) for different brain wave frequency, such as delta, theta, alpha, low sigma, alpha, high sigma, beta 1, beta 2, and gamma activity, which is at the fastest 30 to 70 Hz. Monozygotic twins, the green bars consist shows higher ICC interclass correlation coefficient values across all frequencies compared to dizygotic twins, which is in the pink bars. White matter, this research visually demonstrates the genetic influence on brain function, revealing how monozygotic twins were stronger neural activity pattern than dizygotic twins. Insight from such studies can help advance neurophysiology, brain development research, and medical applications, particularly in areas like cognitive health and machine learning for neural monitoring. So here we see the biological clock, our circadian rhythms, and circadian rhythm is internal biological clock that regulates the sleep-awake sleep cycle over 24-hour period. It's influenced by external clues such as light and temperature. And the genes involved are clock genes, BMAL1, central to the maintenance of circadian rhythm, and also period genes and cryptochrome genes, which regulate the timing of sleep and wakefulness. So we already reviewed the stages of sleep, non-REM sleep, which are non-rapid eye movement, stage 1, 2, 3, and REM sleep, which is rapid eye movement sleep. And the genetic influence by various genes like SLCIA2 and NPSRI have been associated with regulation of different sleep stages. So sleep duration variability varies widely among individuals due to genetic and environmental factors, and there are different factors of the genes that can affect. So the twin study we reviewed showed that sleep patterns have significant heritability, including duration, quality, and timing. And the heritability estimate for the sleep duration range from 31% to 55%, indicating a vital genetic component. The chronotype, morningness or eveningness, also has genetic basis, with heritability estimate around 50%. So we also reviewed the common sleep disorders, insomnia, narcolepsy, and sleep apnea, and the different genetic factors. Many sleep disorders have genetic footing, though they have often polygenic and involving many genes. So we talk about narcolepsy, and we are going to have another class with more detailed narcolepsy, a sleep disorder characterized by overwhelming daytime sleepiness and sudden loss of sleep attacks, which is due to muscle loss. The key genes are HLA complex and strongly associated with narcolepsy, especially narcolepsy with cataplexy, where they have muscle loss. Other genes include TCR, which is a T cell receptors genes influencing our immune response.
Lack lack of sleep may do more than leave us cranky the next day. A new study suggests it may have damaging effects on our genes. Dr. Carol Ash is director of sleep medicine at Meridian Health and joins us now. Good morning.
Good morning.
Okay, so what does this study show?
Well, this is an amazing study. I mean, we've long known at Meridian Health that lack of sleep has an impact on your health. What this study is showing is after 1 week of less than 6 hours a night, it will actually affect the activity of your genes, and up to 700 of them. And genes that control for metabolism, immunity, inflammation, and stress. So this is really a dramatic study.
When you say affect your genes, how does that work, and how does it manifest itself in terms of your health?
Well, Charlie, think of the genes as the instruction manual for the production of proteins. And proteins are very important. They're the building blocks for everything in our body. So if you're changing these genes, they're actually turning some on and off and changing the way they wax and wane in their activity throughout the day. And so it literally will affect your health. Heart attacks, stroke, diabetes, obesity, all these things have been linked to sleep. And this is suggesting a mechanism at the molecular level how that may be happening.
We hear lots of studies about how the lack of sleep is bad for our health. Why do you think this study is different?
Well, you know, again, the other studies that we've seen have all looked at the endpoint. You know, this is what we can see: heart disease, stress, inflammation. We know when you're not sleeping well at night, you see a lot of inflammatory substances in the bloodstream. So finding the actual mechanism gives us an opportunity to look at future interventions. You know, unfortunately, 24 hours a day, 7 days a week, we're all living in a society that really asks us to push past barriers. So having interventions and future treatments is good.
So what are your tips for sleeping better or getting some sleep?
Well, for most of us, I mean, there's 70 million Americans that have sleep disorders. So those individuals, it's a problem. They really need to reach out to a sleep specialist. But for the rest of us, you want to pick the same time to bed and wake up time. So a regular and consistent schedule. Get the right amount. Most of us need 8 hours, and create a relaxing bedtime routine about an hour before bed. And if you do that, Nora, for a week, you should be jumping out of bed in the morning and feel very refreshed.
She already jumps out of bed in the morning.
Dr. Carol Ash, thank you.
So personalized medicine requires tailoring of the medical treatment of a person to the individual characteristic, including the genetic makeup. Genetic testing for identifying specific genetic variants that influence sleep leads to more targeted and effective treatment for individuals. So when we use personalized medicine and identify the genetic variants and help in treatment, that is more effective treatment. Understanding how genetic differences affect drug metabolism can also help customize medication regimen for sleep disorders and in the process of pharmacogenomics. So a case study for a genetic analysis for a patient with a family history of sleep disorders undergo genetic testing. So this patient has a history of sleep disorders, family history of sleep disorders, and that patient goes under genetic testing to identify the main characteristic. So the analysis results identifies a mutation in PER3 genes, which is associated with delayed sleep phase syndrome. So DSPS, which is delayed sleep phase syndrome. The outcome of this study was that patient treatment plan is adjusted based on their genetic profile, leading to improved outcome. So when we do these, when we are testing and making the treatment according to the individual genetic makeup, that will make more qualified and more effective treatment and will have better outcome in sleep. So there are various ethical considerations that should be understood in genetic or testing or sleep disorders. So for having different ethical issues for genetic testing, there's special consideration for the privacy of the patient, because I think everyone knows about the HIPAA compliance. The HIPAA rules, there are multiple identifier, 16, 17 identifier, which, for example, name, date of birth, social security number, address, various factors where you can use them to identify a specific patient. So that has to be kept confidential and should not be accessed by any person or for the any use which is not related to the patient treatment or for the insurance purposes. So there are concerns about the confidentiality of genetic information is critical. Also, genetic discrimination. Individual could face discrimination based on the genetic information. So if you have that information or someone has the information about the patient, about their genetic um, um, information, those patient or people can be discriminated on the basis of that genetic information. The informed consent is essential to ensure that individuals fully understand the implication of genetic testing, including potential risk and benefit for the testing. So when we are talking about the future in sleep genetics, the emerging research areas that may include and can benefit for patient diagnosis and treatment, one of them is CRISPR, and then Cas9, which has a potential for gene editing to treat genetic sleep disorders. If somebody has a genetic disorder, so CRISPR technology can be used for editing genes, gene editing, and help in treatment of disease. For neurogenomics, exploring the genetic basis of brain function, which is related to sleep, can help for the future studies in sleep genetics and for the treatment of sleep disorders. The potential impact of that is that advances in genomics may lead to new treatment for sleep disorders for the patient specific to the specific alterations and a deeper understanding of biological mechanisms for underlying sleep. And so at the end, I'm just going to mention this, that obstructive sleep apnea or OSA. So sleep apnea, as I mentioned earlier, is a condition where the person has difficulties in breathing, and that can be because of two major factors. One is called obstructive sleep apnea, and the second one is known as central sleep apnea. So the difference between that obstructive sleep apnea is the characters of that is can be result of obesity and high amount of tongue fat or large size of the tongue, craniofacial structures which can cause blocking or difficulty in breathing or blocking of the airway, soft tissue structure that can block the airway, neural control of the airway, and the other factors. So there are multiple pathways to obstructive sleep apnea, and each of these pathways are likely involving many genetic variants. These multiple pathways make finding gene variants, conferring risk or protection for obstructive sleep apnea more challenging. So we are going to talk about more about the sleep apnea and these type of apnea in future. So in summary, genetic plays a crucial role in regulating sleep, influencing both normal sleep patterns and development of sleep disorders. Advances in genomics and personalized medicine offer new opportunities for understanding and treating sleep disorders, and the ethical consideration are paramount as genetic testing becomes more integrated into clinical practice in future. So going back to the question number one, we talked a 23-year-old woman with restless leg syndrome is curious about the risk of her passing it on to the newborn baby. So genetic passing on to the newborn baby, and because the baby father also had the same diagnosis of restless leg syndrome or RLS. What are the risk factors for the baby to inherit the disease? So restless leg syndrome, the rate of inheritance, if one parent has RLS, it's about 30 to 50%, and if both parents have RLS, then it is about 50 to 80%. So that's the end of today. Thank you.